Yeah, so I'll be talking today about like a method that we have been developing for like a number of years now for predicting evolution of seasonal influenza lineages, which is based on like a fitness model, like a mechanistic explicit fitness model for circulating strains. Yeah, I want to thank my collaborators, not to forget. So like that's the work has started when I was a postdoc, and I was working with Michael Lessig at the University of Cologne. And now like we have been developing it further in the recent years. We have included the antigenic data component in collaboration with Richard and William, the organizers. Also like in discussions with John Macaulay and Rod Daniels from Francis Crick Institute. And there has been also other like students and postdocs involved in automatizing and like bringing this whole set of tools that we're developing, so that we can really like keep up to date, like keep up with the data and yeah, prepare this like reports of predictions. So we also like yeah, so this method that I'm going to talk about like we have been for a number of years now, like since 2015. Now we have been like preparing reports for the WHO, which is like for the last 3 years. I think it's been more. It has been more. Like we actually been invited to the vaccine consultation meetings as well, and we regularly get data for models from the WHO collaborating centers. All right, so just to talk about the method, so like the questions that we started like when we were working on this method, on this approaches was, can we quantify selection in influenza to predict evolution? What are the limitations of such predictions? And if these predictions could be useful for vaccine strain selection. So influenza, so like in general, the problem of predicting evolution is like, I mean, it's difficult, and influenza seems to be a great system for such for addressing such questions, mainly because of loads of historical sequence data. So we have data in databases that goes over like the last half century, I think by now. It's relatively, one can think there's like relatively, there's some like, it's characterized by some genetic simplicity, of course, like when you compare it to other like other systems in terms of like number of like genome sizes and the number of proteins. I'm not taking away from influenza's complexity at all. But like from like when you compare systems, it seems that it may be more easy to study. So like most of the evolution that is observed in adaptation and genetic variability is restricted to the two surface proteins, hemagglutinin and neuraminidase, and just from analysis of the phylogenetic trees, we see there's like some clear signatures of selection. So here's an example of H3 and 2 tree that spans from like from 2010 to 2020. This is the recent tree. So like what we see is that this tree, well, I mean, if it was drawn even like back further, we could see that this tree is very elongated but it's characterized by a high genetic diversity. So so this every new clade that is like that can be distinguished by antigenic properties or big number of mutations or reassortments is here colored with a new color. And so at each point in time we see, oh, sorry, there's Siri turned on in my phone. So yeah, so at the same point in time we see like many competing clades. So this also, right, so for predictions, it's like very important, right? Which of the clades that we see at a given point in time is the one that is going to take over and be at the trunk. All right, so yeah, so here's actually like, this is like illustration of the prediction problem. So like imagine like we are at a given point in time. So like say it's February, like now, and then we need to know what's going to be circulating for the next, in the next season, in the next winter season for the vaccine strain selection, and say we have a number of competing clades that I distinguish two of them for this example, the orange one and the purple one. So now if we had a fitness model, if you could like assign fitness to these different computing clades, so fitness is a growth rate, right, some expected growth rate, expected number of offspring. So if we knew the fitness of those clades, we could predict. So we could just project how the frequency would change. There's like a simple like equation here. So if you are, if a given clade alpha, so the say orange or purple one has a given frequency at a point in time, then with a fitness model, yeah, we can project those frequencies with time and predict what this frequency is gonna be. Of course, like this model, like so just I think it's gonna be like I just want to like point it out already here that it does not take into account any new mutations. So we only can like ascribe fitness to the clades that are already circulating. Yeah, so like so the validation comes a year later, right? So if we make a prediction, then we can like a year later from like collecting sequence data, we can see which clade indeed grew, and we can compare the predictions to what was observed. I mean, this is of course like just a toy example here, but I'll be showing like more of these cases. All right, so like so now the question is like if you want like that it would be handy to have such a fitness model. So what we thought is like there are two components that should be taken into account in a fitness model for influenza. It would be there's like innovation, the need for the virus to be innovative, to evade the immune recognition, and stability, so that it like still like has the conserved function, that it's still has the proteins are still folding, and that it's still able to that it still maintains all the necessary viral functions. So yeah, so innovation, so like we would like call it this antigenicity. So he like so what we mean here is that right. So here's an example of a I mean we've seen one of those pictures yesterday in John's talk, for instance. But so here's an example of hemagglutinin, of influenza hemagglutinin with an antibody that is bound to a specific part of hemagglutinin. So these are the viral epitopes. So if so recognition by human antibodies decreases fitness of strains. So like these are the human antibodies that the hosts have because of the previous infections or because of vaccination. And then the second component would be like the stability of conserved function. Here I'm like showing an example of like just loss of protein folding. So if we have mutations that happen outside those regions, also those regions actually where this changes would be promoted, and then on average we would expect that they may destroy like viral proteins or make them dysfunctional. So here's some analysis from sequence evolution data that actually like shows that that even from like historical data we see a signal for this like for the separation of epitope and non-epitope mutations. So if you just look on a simple like statistic here, like the number of substitutions and the ratio of substitutions to the ratio of mutations. So substitutions are the mutations that are fixed, that get fixed in the population. So these are the ones that happen on the trunk of the tree. So this is historical like trees, and we can look on the number of on the class of synonymous mutations, which we here like treat as the ones that they don't change function. So there are this is the class the neutral class, the class of non-epitope amino acid changes, which here like we think that they would be on average deleterious or slightly deleterious, and the class of epitope amino acid changes that we think that are beneficial. So even like just looking on this tree, so like we actually highlight the ones that happen on the trunk. So these are the substitution, and you see that the non-epitope mutations are really depleted here on the trunk. So here you see 2 cases in this data, and while you see really many epitope mutations that gets just fixed, and they fix in the population. And
Sorry,
like if you look on the numbers, just yes.
Can you just explain what the axes are on the graph there? It's just too small for me to read. The ordinary.
Oh, sorry. Yeah. So this is actually the number. So this is just the number of mutations. So this is like this is the number of mutations from the trunk. But
Cool.
it's yeah. On the other, like on the more new figures I'm showing, this is usually the time. So that's. But I'll make sure that I tell that. So yeah. So yeah. So what we see also from the statistic is that the number of non-epitope mutations is depleted. It's 0.3 by factor of 3, and the number of epitope mutations is increased. It's the ratio is 2.5 to what you see in the class of neutral mutations. All right, so yeah, so this is like, so this like just drove us so like to this like to so we're gonna like build a fitness model, so like just like to maybe give it some like biophysical grounding again. So if you think that we have so we have affinity between a virus like hemagglutinin here and an antibody. So when like affinity is large, then this the two bind. But when virus acquires mutations in the epitope regions, it will decrease the affinity, which at some point with some like accumulation of this epitope mutation, so sometimes like, it may be that it's just one, but it's usually probably would be more. So like it will lead at some point to loss of binding. But this is, so what I want to point out here is that this is this would be modeled by like this, that we would use this thermodynamic model here that depends on the affinity. So like, 1 or 2 mutations would be fine, but at some point you just lose binding. So it's not a linear function altogether of affinity. At some point like this, also like if you have really quite a lot of epitope mutations, then it stops mattering. So this is like for one antibody and one virus, and then like similar story here for another functions, like on the other side of the fitness model for stability or like conserved functions, that non-epitope mutations may just lead to increase of like free energy of the protein, and at some point with some like accumulation of those mutations, it becomes lethal for the virus. But it again, like it would be happening in some like this non-linear way. So this is like, yeah, so this was like, I was just talking about this phenotypes, like binding probability or unfolding probability. We can translate it directly to fitness. So yeah, so for fitness, so fitness would be when you look on the binding probability here, right? So this is it's not deleterious for the virus. It's not beneficial. It's beneficial for the virus not to be bound. So that's why the fitness will actually come with like a negative sign over here. And then there's like, we also will attribute some weight to this component. And this is something like, this is a model parameter, right? So this is how much this component weighs. This is something that we would that is get to be trained from data. And then similar consideration here for the other component. This is also, we will just translate it directly into fitness. Yeah, with some weight that will be assigned to this component. We also will decide what is the threshold really that's where we see like how many non-epitope mutations really leads to this loss of, well, like this change of the phenotype and fitness.
Marta, do you mind very much if I ask another quick question for the
Sure.
physicists who aren't in your field? Can you just say very quickly what you mean by epitope and non-epitope?
Oh, sorry. Yeah. So of course, so epitopes are these antigenic sites there where the antibody binds. So and we know in influenza, so we talk about this like specifically 5 epitopes. So A, B, C, and A, B, C, D, and E. And they are even like not, these are not linear epitopes. They're really like just sites that are close in 3D structure, one next to the other. So, and they are known, so we know them from, I mean, okay, so the list is actually some like people like still like converge on them, and they may change, but like we here we just used a published list of those sites.
But they're sites on the viral protein.
Exactly, in hemagglutinin on the head, in the head domain.
Sure, thank you.
Okay,
And
sure.
non-epitope is everywhere else. It's internal to the virus. It's sites that do other things.
Yeah, and they are like predefined.
So great.
So yeah, so like these are the sites where you like expect to see antigenic differences. All right, so like yeah, so now like when we have these two components, like you know, the fitness would be like just the sum of the two. And I like talk about like this antigenic component. This depends on time, and I will like get to say why. So like the same virus in a given point in time, like this year will have different fitness than next year, which has to do with the fact that like just more that the host population is changing, and so it actually requires escape. All right, so like just like to like how we can really implement that, because like those affinities or free energies that I was talking about, they are not like we need the measurement for that. So what we started with like first in 2014 was that we used sequence distance as a proxy for binding, like for the affinity between a virus and an antibody that has been like created in the host in response to previously circulating viruses, to previously circulating virus. So we here like assume that like, yeah, if I have virus I, then there has been, yeah, in the past there has been virus J, and but yeah, and then the affinity or like between them would be proportional to the sequence distance between the two, and actually not sequence distance, but the number of mutations in the epitope region. So that's what we've used, and that's so we use this sequence distance as a proxy. And then for stability, we actually like also use a very simple measure, and then we actually, this is what we still use. We just counted the number of non-epitope mutations. So and then apply the nonlinear like transformation on this on the number of accumulated non-epitope mutations. All right, so so this was our first approach to that, and since then we actually started, and also this was done with collaboration with Will and Richard. We started like incorporating like better measurements of from hemagglutinin inhibition and the neutralization assays. We've heard quite a lot about this yesterday already, but in principle, so those assays measure like tighter reduction between like a reference virus that a ferret has been infected with and a new test virus. So again, so like high titer means that the two viruses are similar, and then low titer means that they are dissimilar. And then like application of this data, like it allows us to really go away from the sequence model and have a better measurements. So and this is what we're going to be using like now in like what we're using now instead of sequence. So this is how the data, the typical data looks like. So we have this is data from Crick, an example that so there's like number of reference reference viruses that have been raised in ferrets, and then the test viruses here. Those viruses belong to different clades, genetic clades. So one can like see like how like those measurements can act as some like, replicates. But by no means like this data is complete. So this means that very often like we actually deal with like, so we don't have like every possible reference clade measure against every virus. So yeah, so this is like some way, like some view of the data that we have when we like after like we even average over the genetic clades. So these are measurements here that show tighter reduction. So here like low value means that the reference virus from that clade is recognizing well a test virus from that clade well. So you see, like there's like a lot of virus. There's like quite a lot of measurements here, but there's also some missing. And here I'm just listing in a set of clades that have been like considered this year in H1N1. So there's like a need like to fill in to fill in this gap so that we can apply our model, which I'll just say talk in a second. So one more thing that we so that's so like one thing that you explain to see here is that there are two clades, two groups of clades. So there's some additional mutations here, but you see there's some dichotomy here, that there are some clades that are well recognized by those viruses, but there's another family, another competing clade that is completely not recognized. All right, so we have proposed some an inference algorithm that fills in those gaps. It was based on like interpolation on close measurements on the tree. I'm actually not going to talk much about that because I wanted to actually explain more how we then use those measurements. But it lets us like sort of fill in all those gaps and so that we just can compare every reference clade and every test clade, every test virus. So because like I was like so far already talking about like what happens like when you have one antibody, like one reference virus on one reference clade and one test virus. But what is really like important here is that actually we have like high viral diversity, and what a host sees is really like a bunch of viruses, and this is really what so a given host may see different viruses, and that's sort of like what is important for like we need to know what this host will be susceptible to. But the same thing is on the virus side, that it actually is the viruses are probing many different hosts, and those viruses that can infect more hosts are going to be more successful. So there's some of it's a bit of a co-evolutionary story, right? So the antibodies of the host populations are keeping up with the evolution of the virus that is escaping the immunity of the host population. So how it would, so now I want to show what the complete fitness model looks like, that takes into account this host population diversity. So what really matters for a given virus I, right? So this is a given virus strain. So this would be a tree. So like say we consider a virus somewhere here. So we know that its fitness will be decreased if it's similar to the previously circulating viruses, but the weight that like the penalty that comes from that will be proportional to the fraction of the population that has seen the previously circulating virus. So what we need here is like we need to sort of stratify the host population into somehow infer what is the structure of the population, and how infer what are the proportion of this individual cohorts, immune cohorts. And what we do is that we think is that we approximate the size of such cohort that has seen like previous viruses. So here, like we know, we say like we noted with raw, that this size would be proportional to the number of infections that we've seen with this class of viruses. So like the tree that we like see here, like it not only represents the evolution of the virus, but we also use it to count the number of cases, right? So and like just estimate how big this cohorts are. And then for every virus I here and every cohort raw, we have we have here this proxy for the affinity of antibodies against this clade raw and virus I. And this we have from this antigenic measurement, also all from the sequence model. We applied this nonlinear transformation on that, and this like gives us the binding probability, the binding probability. And then the viral fitness will depend on basically on the average of the over the immune environment. So yeah, so we just compute, we weigh similarity of the virus to every previously, like to every immune cohort by the size of this cohort. So this is sort of the total like way how we like just sum over those over the history of the viral population. So here's like an example like how this is implemented on the data, right? So this is this table that I've been showing before. So here we compute a fitness for every row of this matrix. We sum over all columns of the matrix. The raw is the weight of the cohorts. It's just this would be like proportional to the number of cases we've seen with this clade. So this we can like estimate just from counting. So this is like also denoted here with color. So darker colors mean higher weight. And then we take just the inferred value of the titer and like just properly translated like with this with our model, and the parameters and the shape of the curves are being fitted to the data. So that's so this is like so that sort of I went into the details of this immune component. So if we have like that, so we systematically like do these predictions like every year. The model has been like evolving from the sequence model to this antigenic model. So here I'm showing like the predictions with the antigenic model, some like examples, like for example in 2013. There were like two main competing clades. So we predicted quite well. You sometimes see that you may see some divergence at some point, which could also be attributed to just that there are new mutations that are appearing in the population. So that's this is prediction 1 year ahead. But as I was saying, we do the predictions only for the clades that are circulating at a given point in time. This is another example also from the past. This is an interesting example because that was the rise of clade 3C2A that actually was not caught at that time. So I'm showing that the model would have predicted it well. However, it would not have because there was no sequence data available. So this was actually the clade that was, this is February line. There was very few sequences available back then, and I think this sort of like this situation has also led to like some increased sequencing efforts. So like since that time, the number of available sequences has really improved. All right, so then sorry, I'm just in the interest of time, I think I want to also show an example from an interesting example from H1N1 from last year. So H1N1, you see that it actually has been not showing so much of adaptive evolution for a while, but then since from time to time there was a mutation in position 156K that was significantly changing antigenic recognition of the clade, and it was appearing quite often, and then but maybe due to some not other components, it was actually not staying in the population until last year. where like we've seen a number of strains already like in January. So here's the tree that is colored by fitness. So like right is like red is high fitness. So given this this fitness at this point in time, we had made such a prediction. So this is the purple line. Here's the clade. So we have been seeing this in end of January or in February at around 10%. And we were like, predicting that that it will take over within a year. But in particular that in April, this is April, it will reach 50% of the population, winning over this other clade that was also circulating over there with, it's called like 187A clade. And that's what like happened after that. It's actually until April, and indeed this clade reached 50% and was winning over the green clade. But then the lockdowns happened. And so actually now at this point in time, sort of the evolution kind of was like everything was stopped. But what we see, like we see really small numbers in H1N1. There was altogether 28 or 27 sequence isolates. This clade was actually like dominating, but it was represented by 18 sequences from a local outbreak from Togo. So it's very difficult to say what's happening over here now. All right, so yeah, so this is just like saying that, but this is very likely because this clade is still over. It's gonna be back on track, and it's also gonna have again this strong fitness advantage, and it's this very fast growth. Okay, so like just my last story is like just how this can be, all this thing can be used for vaccine strain selection. And here's like a very similar story that what we've done for fitness inference and for assigning fitness of viruses. The problem is just transposed. So what we want to do for vaccine strain selections, we want to choose one vaccine candidate from these reference columns here that will have the best protection, right? That will have the best will show the best cross reactivity over the viruses. So what we want to think is that, like, if you look on the frequency of the clades, and here you like, this is where the predictions are handy, because you want to use the frequencies as predicted in one year, like into the next season. So you want to maximize the protection, like the cross-reactivity of this vaccine virus against the future population. So this is like just a simple equation that we use. Okay, so like we again have a vaccine strain is somewhere from one of these columns over here. We sum over clades alpha, that are the rows of the matrix. We weigh it by the frequency of the clade, the predicted one. And then we use the cross-reactivity measure that comes from this, that is based on those measurements. And this is like, so this is an example how this can look like for the individual different candidates. We can compute this average population cross-reactivity for tracking, like using tracked frequencies in the past. And then we can also predict what they will look like in the future. So this is an example, like again, like this figure has also is an example from 2019, from September, for ranking different vaccine candidates. So we like back then we have marked Switzerland 8060 2017 as the vaccine candidate, and so actually got the one that was selected. And yeah, so here like this gray ones are the actual vaccines, and while the yellow ones are the that would be suggested by the model, by this ranking. Yeah, so this like just like to show that this is something that like where we can we use the predictions to rank the vaccine candidates, and we can also what we can also do here is that we can also like assess how much the predictions really affect the ranking. So it's not necessary. Sometimes there may be a clade that is not really, there may be a virus that is not really from the growing clade, but may show good cross reactivity and be and it may be robust. The prediction for vaccine strain selection and the ranking may be robust to exact evolutionary dynamics and predictions. All right, I think I have to maybe I'll stop at that in the interest of time, and I can take questions if there's time for that.